everyone, it's me Amanda and welcome back to my classroom. Today I will be teaching Biology Form 4 Chapter 3 about plasma membrane, subtopic 3.2, the concept of movement of substances across a plasma membrane. So in this subtopic we will learn three things, which are the characteristics, passive transport and active transport. Okay, so we will go into characteristics first. Okay, according to the textbook, there is three characteristics, which are molecule size, polar molecule, and ionic charge. So the first one are non-polar molecules. The first one is non-polar molecules. The second one is small molecules and ion. And the third one is large molecules. I will repeat again. The characteristics of the substances that can go across the plasma membrane is, first one, non-polar molecules, small molecules and ion, and large molecules. So the examples of non-polar molecules are fatty acid. Okay. The second one is glycerol. The third one is fat-soluble vitamins, which we learn in Form 2, which is A, D, E, K. And lastly, steroid compounds. Okay, there are four non-polar examples of non-polar molecules. There are more, I think. Fatty acid, glycerol, fat-soluble vitamins, which is vitamin A, D, E, and K, and steroid. Steroid is a type of lipid or fat, which we will learn in Chapter 4. Next, examples of small molecules and ions. The first one is polar molecules. One example of polar molecules is water. Okay, is small molecules, by the way. Okay, another small molecule is but this one is non-polar. Examples of non-polar is got oxygen and carbon dioxide. Okay, and lastly is ion. So examples of ions are potassium, sodium, got calcium, magnesium. So those are ions we learned in chemistry. So for the second group is small molecules and ions. Small molecules are polar molecules and non-polar molecules. Example of polar molecules are H2O, and non-polar is oxygen and carbon dioxide. This is the one I talked about in my last video that can just go through the phospholipid bilayer. This one is the one that needs to use carrier proteins because it's way too big. So for large molecules, example is glucose. And another example from here is amino acid. Amino acid is the protein. So this one needs to use the protein channel. So I'm gonna repeat again the characteristics. Non-polar molecules, fatty acid, glycerol, fat-soluble vitamins, steroid. And then small molecules. Okay, this is small. I know this is non-polar, but it's small, so it's considered as small molecules. Polar molecules, which is water. Non-polar, oxygen, carbon dioxide, they are gases. And ions, which is example potassium, sodium, calcium, magnesium. And lastly, for large molecules, glucose and amino acids. Okay, next in the subtopic is about passive transport. So passive transport, there is three: simple diffusion, osmosis, and facilitated diffusion. So basically, passive transport is a transport uh, at the cell membrane that does not use energy. No ATP is used. Okay, so the first one is simple diffusion. So we've learned about this already actually. So simple diffusion is the movement of molecules or ion from a high concentration to a low concentration. The moving molecules are said to move down the concentration gradient until a dynamic equilibrium is achieved. So let me draw a little diagram. Okay, let's pretend this is the plasma membrane. Anyways, so this is the plasma membrane, okay? And this is the molecules, okay? There's 8 molecules here. This is the outside of the cell, by the way. Okay, this is the outside of the cell, this is inside of the cell. So this is the molecules or ion. This one is inside of the cell. This one also the molecules or ion. So you see, it's so obvious that outside of the cell, it has even more molecules. It has even more molecules here. And inside of the cell, there is less. So they want the dynamic equilibrium to make it equal. So the molecules will move following the concentration gradient from a high concentration area to a low concentration area to achieve the dynamic equilibrium. So it will move here one by one. There, it's already equal. Okay, it's already equal. So usually they will just keep going. Okay, that's all for simple diffusion. Osmosis is literally the same thing with simple diffusion, but it uses water. So let me draw the little diagram again. Okay, so this is outside of the cell and this is inside of the cell. So I'm going to draw the water molecules. Okay, this is water. Okay, osmosis is the net movement of water molecules from an area of high water potential to an area of low water potential. So basically, it's water molecules moving from a high concentration area of water to a low concentration of water. So there's more, there's less. They want to reach the dynamic equilibrium. So they will move through the plasma membrane from high to low. But then for osmosis, there's also another thing, is that they have solutes. So let's say, usually if there is more water, there is less solutes. And then when there is less water, there is way more solutes. You see? So osmosis, you can describe it to, which is water going from high concentration to low concentration, or water going from low concentration of solutes to high concentration of solutes. Also to do the same thing to reach the dynamic equilibrium. Just now for simple diffusion, they follow the concentration gradient. This one also follows the concentration gradient because it goes from high to low. Okay? And the last one for passive transport is facilitated diffusion. Facilitated diffusion also follows the concentration gradient to reach the dynamic equilibrium, but this one, they need the help of the proteins, the channel and carrier proteins, like the one I said in the last video. Even though this one uses protein, it still doesn't use energy because it's under passive transport. Okay? So let me draw a diagram. Okay, so for facilitated diffusion, like I mentioned just now, they use the 2 proteins we learned in the last video, which is the channel protein and the carrier protein. So the channel protein forms channels or canals to allow small sized solutes and ions to diffuse across the plasma membrane. Channels have specific internal characteristics that only allow specific ions to pass through it. So basically, the ions or solutes, they will just pass through it. It's like a little tunnel, you know. Yes, a tunnel. But then for carrier proteins, carrier proteins have specific sites and can only bind to a specific molecule. Okay, so carrier proteins are specific. It can only use for specific molecule. Okay, for example, glucose. So glucose can only bind with this one protein. Okay, this is a glucose carrier protein. So it can only bind with that carrier protein. Other carrier proteins cannot carry glucose, and this carrier protein, the glucose carrier protein, cannot carry other molecules. So how does carrier proteins work? Carrier proteins, like in the last video, it's a door. So let's pretend my hand is a carrier protein. Okay, so it will open first, and then the molecule will go in, and then will close, but then the inside will open. So that thing will just keep repeating slowly. I don't know slowly or fast now, but as long as the molecules can go into the cell following the concentration gradient to achieve the dynamic equilibrium, then that thing will just keep going in and keep diffusing in. So that is all for passive transport. So now we're going to go into active transport. Okay, active transport. Active transport, so active transport uses energy. They use ATP. Why? Because it goes against the concentration gradient. So just now for passive transport, let me draw a little diagram. So just now for passive transport, they will go from high concentration to low concentration. So that one follows the concentration gradient. But for active transport, the molecules or the substance going across the plasma membrane, it goes against the concentration gradient. That means it's going from low to high. So if it's against, then it's really hard for it to move, So it needs the help of carrier proteins again. Yes. They need help from the carrier proteins, and they need energy because they need energy to move to the higher concentration, because the higher concentration is high. So they need energy to go into the high part. Okay, I hope you understand. So one example from the textbook is a sodium potassium pump. The sodium potassium pump is actually really famous. So I'm going to explain about that. Okay, so now I'm going to explain about the sodium potassium pump. It's in the textbook page 55. So the first things first, this is the carrier protein for the sodium and the potassium. So the carrier proteins that are involved in active transport, they are called pump. Okay, they pump it out. This sodium potassium pump obviously will involve the ion sodium and potassium. So first I'm going to explain about the sodium. So the carrier protein will first open in the inside. Okay, so the sodium, they will go to here, sodium potassium pump, they will go to the protein, then they will attach itself here. Oh my god. So usually the sodium part has 3 and the potassium has 2. So this is potassium, so we don't care about this first. So they will go parking there. And then there's this thing called ATP. So ATP is the thing that makes energy. I mean, it's the energy. So the ATP, it will break. ATP looks like I don't know how to explain. If you want to know more about ATP, you watch Amoeba Sisters. They made a video about ATP, so they will explain to you about that. But then ATP, they will turn itself into ADP. How come ATP become ADP? Because this one T got 3, D got 2. Okay, they will break itself. They will separate itself, become ADP and another P. Okay. So this one have 3 P's, this one have 2 P's. Because one of the P's go out. So this P, they will go here. Okay, they will go to the protein, give the protein some energy. So the protein will shake, shake, shake and move. So what happened to the protein? The protein will open to the outside. Okay, so the carrier protein is open, and then the sodium will go out to the place where there is high concentration of the sodium. Okay, so now I'm going to explain about the potassium pula. Okay, so you see there's more potassium inside the cell. So the potassium, it will go to the carrier protein that will stick itself here. And then this P from the ATP, it will move. It will go back to the ADP and then become ATP again. So it became ATP again. This one has no more energy. So this one will shake, move, and then it will close back to the inside of the cell. Okay. So the carrier protein, it will open back, it will close outside and then open to the inside. And so this potassium can go into the cell. Seriously, that is all for sodium potassium pump. And that's all for 3.2. Thank you for watching.